Hi students, can you hear me? Please let me know if you can hear me so that we can start the session. Hello, can we start? Great. Hi students, can you hear me? Just a second. Please let Okay. Great. So hello, I'm Dr. Devyani Puri and welcome to another live session with me. And today's topic is going to be about staining techniques. So the first thing that I want to talk to you about is that why staining is required, right? So we all know that the bacteria are actually translucent, right? So when bacteria are translucent, we are unable to see these structures very nicely on your light microscope, right? So because of that, because they are translucent, we are unable to see under light microscope. Right, so when we are unable to see them under the light microscope, so we have to do certain things for them to be visible, for them to be more visible. Right, so what do? We have to increase the contrast. And one way of increasing the contrast is by using certain staining techniques. Right, so for the organisms to become visible, so we use certain staining techniques. So let's see about this picture right here. Can you see the picture on the left? These are the unstained specimen, right? And these are the stained specimens. So what happens is my voice echoing? Can somebody tell me if my voice is echoing? Hello. Okay, I'm going to continue. Just a second. Okay, so the ones on the left are the stained specimens, and the ones on the right are the ones on the left are the unstained specimens, and the one on the right are the stained ones. So can you see these exact same bacteria when stained have become so much more visible, right? So staining is important because one, we want to see the bacteria, and secondly, we want to see the uh, just a second. Is it still echoing? Okay, I'm going to continue. So the ones on the right are the stained specimens, so we are able to see the bacteria more, properly on the light microscope. And can you see we are even able to appreciate the internal structures? So we are able to see the internal structures also, right? So that is why staining is very important. Right. So next that I want to talk to you about is fixing of a specimen. So what happens is that before we start any kind of staining technique, we are supposed to fix the specimen. What does it mean? That whenever we make a specimen on a slide, right, we don't want that specimen to get washed off. So we don't want it to get washed off when we are trying to stain it, right? So because of that, what happens is that we have to fix the specimen. Secondly, we have to fix the specimen because we want to preserve the internal structures. So these are the two reasons why we should fix the specimen on the slide, and we have devised certain techniques to do it. So what are the three techniques that we do? The first one is the air drying. So the first one is the air drying. So what we do here is we have just taken this slide and we have a smear on it, right? So we have just taken the slide and we have a smear on it, and we just put it in a diagonal way. We just put it in a diagonal way in some point that the water trickles down, right? And when air passes around it, it dries the slide. So that is one way of doing it, which is called as air drying. So now what is the problem with air drying? The first problem with air drying is that it takes too long. So obviously we just have kept the slide, we are not doing anything, air is passing, so it's going to take a very long time for the sample, so a specimen to dry out, right? And the other thing is that the smear, the specimen, is not fixed properly because air is not doing anything differently to fix the specimen, it's just drying out the specimen, right? So that is why these are the two disadvantages of air drying, that it takes too long for the specimen to air dry, and it takes the specimen is not fixed properly, right? So that is the first one, which is air drying. So then we devised two different methods, the second one being the heat. So what do you see here? This thing right here, what do you see here? This is known as flaming. So what are we doing here in flaming? We are just taking the slide which has the smear on it and we pass it 2-3 times over the flame. Okay, so when we are doing this, what is happening is this heat is going to dry out the specimen and it's going to fix the specimen on the uh, slide, right? But this heating also has certain disadvantages. And what are the disadvantages with this? That if we overheat, so if we overheat, what happens because of that? The proteins or the internal structures that are present inside this smear or the bacteria that is there is going to get denatured, right? So these internal structures can get disrupted. So overheating can disrupt internal structures, right? So that is why this method is also something that we are very routinely using, but we recommend that only try to pass this slide only 2-3 times on the flame and that's it. Right, so that is your heat method. And the third method that we are talking about is going to be the chemical method. So what do we do here? The slide that has the smear on it, we just immerse it under certain chemicals, right? And we leave it for some time. And what are the most common chemicals that we use? We use glutaraldehyde, right? We use glutaraldehyde, we use acetic acid, and we use mercuric chloride. Right, so these are the three things, three most common chemicals that we use to fix the slides. And the advantage of this is what? So what are the advantage of this? So advantage is that it will preserve internal structures, right? It will preserve the internal structures and they can also be used for samples that have blood. So blood smears. So that is why this is very, very important. So there are three methods of fixing. Now we've spoken about why we want to fix this slide, right? Because we don't want to get the smear washed off when it's stained. Because when we are staining, we are using so many chemicals and decolorizers and water and everything. So this smear can get rubbed off. So we have to fix. And because we want to preserve the internal structures, right? So this is what is fixing of the specimen that means. So now let's come to the common stains. So what are the most common stains that we used? First I'm going to start talking about the simple stains. So what are simple stains? Simple stains are nothing but they are just basic dyes. So they are just basic dyes. So what will it do? It will simply stain everything on the smear. Right? It doesn't matter if it's a foci or a bacilli or whatever it is, it doesn't matter. These simple dyes, they will stain everything. So what will simple do? They will stain everything. Right? So these are what simple dyes do. And what are the examples? There are mainly two examples. One being the methylene blue, right? And one other one is your basic fusion, right? So can you see here? It doesn't matter if it's a foci or a bacilli or anything. What is it doing? It's staining everything equally. So this is what simple dyes do, right? Next, we are going to talk about negative staining. So what is negative staining? Negative staining does not stain the specimen or it's not going to stain the bacteria. What is it going to do? Can you hear me now? Is the audio better? Is the audio better now? Can somebody please tell me? Is my audio better? Please tell me. Okay. So I'm going to continue. So what is the negative staining doing? Negative staining is actually staining the background. So it's not staining the organism, it's going to stain the background, right? So it stains the background, right? So can you see here? The background is getting stained and the organism is being illuminated. So this negative staining is actually used for two types of organisms. One is the one that is capsulated, so that we are able to see the capsule better, and the other ones that are too thin, the bacteria that are very, very thin, like the spirochetes, right? So can you see here? So what are we seeing? We are seeing the capsule here and the background is stained as black, right? And here can you see what are these? These are actually your bacteria that are your spirochetes. So spirochetes are actually very, very thin bacteria, right? So we are unable to see these bacteria by naked eye. So we have to use certain stains and this being the negative stains. So that is why it's going to stain the background and it's not going to stain the organism. So what are the examples of negative staining? The most common example that we use is the India ink, right? And the second one that we use is the nigrosin stain. So these both stain the background as black. And against the black background, you are able to see organisms visible, right? Brightly visible. So that is your negative staining. Third, I'm going to talk about the impregnation staining. So what is impregnation? Impregnation is nothing but it's able to, so we are basically using certain stains that's going and adding on to the bacteria, right? It's adding on to the bacteria. Look right here. Can you see this bacteria was hardly visible? So what happens? This stain, it keeps going and adding and adding and adding. And what happens eventually? It makes the bacteria visible, right? So these are known as the impregnation staining, that these stains, they make a layer on bacteria and make it thick so that it becomes visible, right? And the most common stain that we use here is, the most common stain that we use here is silver impregnation stain. So that is the most common stain that we use here. So either we can put it on the whole bacteria and make the bacteria visible, or sometimes we can put this on certain structure of the bacteria, like the flagella, maybe the flagella. Because flagella is also very, very thin of the bacteria, right? It's not usually visible. So these flagella can, these silver stains can also go and attach on the flagella and make it thick and make it visible. Right? So this is impregnating or like putting something on an organism to make it visible. So this is an impregnation stain. Next, I'm going to come to differential staining. So what do you mean by differential stain? So differential stains are something which either differentiate between two bacteria, right? The first thing is that either they will differentiate between the two bacteria or they will differentiate between two structures of same bacteria. Right, so this is what differential stains mean. Either it will identify two bacteria differently, or within one bacteria, if there are two structures, it will stain that differently. So this is what is known as differential staining. And here we are going to talk about three stains in particular. So what are the different types of differential stains that we know? The first one being the Gram stain, right? The second one is the maybe acid fast stain, right? And the third one being the Albert stain. So these are the three most common differential stains that we have invented so that we are able to identify either two different bacteria or different structures of the same bacteria, right? So let's come to Gram staining. Let's start with the first stain, which is the Gram staining. So what is actually Gram staining? Gram stain was actually invented by Hans Christian Gram. Right, it was first identified, it was first invented by Hans Christian Gram. And what did he say? He said that he was able to differentiate between two bacteria. And how was he able to differentiate? He said that bacteria can either be gram positive or they can be gram negative. Right. So he said कि with my strain, my bacteria are going to be either gram positive or they're going to be gram negative. ठीक है. So what did he do? Let's see what all things do you require in a gram stain. So what are the different stains that you require in a gram stain, right? So the first thing, which is known as a primary stain. So primary stain is actually the first stain that is going to stain everything. So this is the primary stain. And here, what is the primary stain that we use? We use crystal violet. So we use crystal violet here, right, as a primary stain. So then second thing is your mordant. So mordants are those things that actually help and make a bond with the primary stain so that the bacteria can retain this stain. Right. So here we use iodine as a mordant. So what it does, it makes a bond with primary stain, and because of that, what is happening? The organism is able to retain the primary stain. Right? So that is what mordant is. Then third thing that we use here is the decolorizer. So what are the different decolorizers that we can use? We can use two types of decolorizers in gram staining. One being your acetone, right, and other being your ethyl alcohol. So these are your decolorizers. So what does a decolorizer do? So some bacteria that are unable to retain this primary stain, when we are putting a decolorizer, it's going to wash off that primary stain from that organism. Right? Some bacteria cannot retain the primary stain. So decolorizer is going to decolorize or wash off the primary stain and they will become unstained. Right? I'm going to talk about the steps in detail. So just first wait with me and just know what all is required. Right? And then we use a counter stain or we known as a secondary stain. Right? So counter stain or secondary stain. And what is the stain here that we use? We use safranin. So safranin is pink in color and your crystal violet is obviously violet in color, right? So saffron is your secondary stain. So all the organisms that were unable to retain the primary stain now are going to retain the secondary stain, right? So that is the counter stain. So now let's talk about the gram staining. Can you see here what are we doing here? Yeah. So this is the first specimen, right? We have made a smear and this is what our smear looks like, and we have fixed it. So we have fixed it using any of the three methods that we were using. So this is what fixed stain is, right? So now in gram stain, what are we doing first? We are going to add the primary stain, which was what? Which was crystal violet. So what is going to happen? Everything is going to stain violet, right? Doesn't matter if it's a cocci, doesn't matter if it's a bacilli, everything is going to be violet. And here we do it for 1 minute. ठीक है 1 minute के लिए we're putting crystal violet on the side, leaving it. So all the bacteria are going to retain the stain. So now next what we are doing, we are going to add iodine, which is the mordant. So now what is this iodine going to do? This iodine is going to go inside the cell wall where the stain is and it's going to make a bond with the stain, right? And it's going to fix the stain inside. So that is why it is a mordant. So we have added the iodine and we have done it for 1 minute. Wait, making, giving time to the iodine to make that bond. So now what we are going to do, we are going to add the decolorizer. And what is the decolorizer here? Either acetone or ethyl alcohol. Right? So we are just going to put do it 1 to 3 drops only we are going to put here. So what will happen is all the organisms that are unable to retain the primary stain, they are going to be decolorized. Can you see these organisms have become decolorized because they were not able to retain the primary stain. And with the decolorizer, they are washed off now. So now what do? We add a counter stain. And what is the counter stain here? It is safranin, which is pink in color, right? So now what is happening is, can you see all these organisms have retained the primary stain? So this is your gram positive. These all are your gram positive. So gram positive are purple or violet because of the crystal violet, right? And all of your gram negative ones. So gram negative ones are pink because they were able to retain the secondary stain, right? So now I'm going to show you, see, this is what your gram negative and gram positive organism looks like. So all the gram negative ones are pink and all the gram positive ones are purple, right? Or violet, whatever you want to call it. But why is this happening, right? So there has to be something different in the gram positive and the gram negative organisms, right? So what is different here? Let's look at their cell walls. So these are the cell walls. So this cell wall belongs to the gram positive organism. Why? Because what is there, if you've seen my morphology video, what is there in the gram positive cell wall? It has a very thick peptidoglycan layer. Can you see this purple structure? It has such a thick peptidoglycan layer. So it has a thick peptidoglycan layer. Because of that, what is happening? When you're adding a decolorizer, the bond of the iodine and the crystal violet is so strong that whenever you're adding a decolorizer, this peptidoglycan wall is so thick that the stain is taking a lot of time to move out. Right? It's almost not able to move out because this peptidoglycan layer is very, very thick. Right? So this layer is thick. So that is why the gram positive organisms are able to retain this primary stain. So gram positive ones are able to retain the primary stain because of this thick peptidoglycan there. Right? Now let's see this structure. What is this? This is a cell wall of a gram negative organism. So what is different here? Look at the peptidoglycan here. So this peptidoglycan here, what is happening is, can you see it's so thin in comparison to this gram positive cell wall? This peptidoglycan layer is so thin. So what is happening? When we're putting a decolorizer here, the iodine and di bond is very, very strong. But because the peptidoglycan layer is so thick, the stain is able to move out, right? The stain is able to move out. So because of the thin peptidoglycan, it is unable to retain the primary stain, and that is why what it does, it retains the secondary stain. So whenever it retains the secondary stain, so what is the secondary stain? It is safranin, and that is why it is pink in color. Understood? So what is happening is gram positive cell wall, thick peptidoglycan layer. Because of that, it is retaining the primary stain, which is your crystal violet, right? Which is your crystal violet, and that is why they are violet in color. And because of the thin peptidoglycan layer, they are unable to retain the primary stain. So they retain the secondary stain, which is safranin, and that is why they are pink in color, right? So that is the reason of the gram stain. That is how that's this stain is able to differentiate. So now I want you to remember one thing, and please mark this somewhere because there are a lot of questions that come on this particular thing. So just remember it like this, that all cocci are gram positive except, so just remember this mnemonic, all cocci are gram positive except three of them, M, N and V, right? So what is M here? It is Moraxella. N is for Neisseria and V is for Vionella. So these are just the three organisms that are gram negative cocci. So these are gram negative cocci, right? So M and B, all cocci are gram positive. So you don't have to remember which cocci is what. Just remember this mnemonic. Just remember the except M and B, right? So this is how you're going to remember. These are the gram negative cocci. So next, one thing I want to you to remember is all bacilli are gram negative, right? Except Is it still echoing? Is my voice still echoing? Is it still echoing? Please somebody tell me if it's still echoing or not so I can continue. Just a second. Let me check. Hello? Can you hear me? Is it echoing now? Hello? Can you hear me now? Hello? Is it okay now? Great. So, okay. Back to Okay, so back to this. So just remember it like this that all bacilli are gram negative, except it's a very, very famous mnemonic that is there. And what is that? It is McDonald, right? It's a very famous mnemonic. I think a lot of people use this mnemonic to remember it. So just remember all bacilli are gram negative, except McDonald. So what is M in this? All the mycobacteria, right? A is for your actinomycetes, right? C is for Clostridium species. D is for your diphtheria or Corynebacterium, right? Corynebacterium. O N, read it ulta, read it as nocardia, right? Read it as nocardia. Then A is for your anthracis bacilli or bacillus anthracis, what you call it, right? L is for listeria and D is for your diphtheroids. So just remember this, write it somewhere. This is just very, very important. All cocci are gram positive except M and B, right? And all bacilli are gram negative except the McDonald. This way you're never going to forget, and you get a lot of questions on this. This both things are very, very important. They will just give you a bacilli and say if it's gram negative or gram positive, or if they'll just give you a coca and say is it gram negative or gram positive. So just remember these two mnemonics, right? Great. So now, next we are going to move on to the acid fast staining, right? So acid fast staining, why do we do it? So there are certain organisms that are weakly gram stained. So because they are weakly gram stained, so what happens because of that? We have to devise certain other methods, right, for them to see their staining. And okay, I'm going to talk about what is acid fastness. So acid fastness is actually the ability of the organism to resist, resist decolorization by some acid. Right, so acid fastness means that it is the ability of the organism to resist the decolorization by the acid. And what organisms are acid fast? Let's see the structure right here. So this is the structure of the acid fast organisms, and one thing which is leading to this acid fastness is this structure right here, which are the mycolic acids. So all these acid fast structures have actually this mycolic acid, and the more the mycolic acid, so more mycolic acid, right? More mycolic acid means more acid fast. That means more acid it's going to resist. ठीक है. So here the most common acid that we use is H2SO4, which is your sulfuric acid, right? So acid fast staining mein we use sulfuric acid. So I'm going to tell you the two most common methods that we use here. One is your Zn stain, right, which is your Zeal-Nelson staining, right? And the other acid fast stain that we use is your Quignon's stain, right? So these are the two, and I'm going to talk in detail about it, right? So Zn staining and Quignon staining. So before I go there, I want you to learn what all organisms are acid fast, because you do get a lot of questions. They will be either picture based or they will directly ask you what are the organisms that are acid fast, right? So let's see here. So I'm going to divide it into three. So either they can be bacteria, right? They will be parasites, and then there will be others, right? So what are the bacteria that are acid fast? So how are we going to remember it? LMN. Like in a sequence, ABCD mein LMN comes in a sequence. So L. So what is the L here? It is the lepra bacilli, right? Mycobacterium leprae. It is the lepra bacilli, and it is Legionella mycardidae. Okay, so this is one, which is the L. Then we have M, which is your mycobacteria. L, M, and N. So what is the N here? N is the nocardia. So this is your L, M, and N. Okay, so this is your element bacteria that are acid fast. Now let's come to the parasite. Parasites, we are going to remember it as CME. So C. So what are the C? C are the coccidian parasites. So which ones are the coccidian parasites? They are the Cryptosporidium. They are the Cyclospora, and they are the Cytoisospora. Right? So these are the C. Which ones are the coccidian parasites? So C. Then you're going to remember it as M. M is for your Microsporidia. So Microsporidia also a parasite. It comes under acid fast organism, right? And then you have E. So you're going to remember two eggs here of a parasite. So what are the eggs? There are two eggs, that two organisms which start with S. One is your Schistosoma mansoni, right? And then you have your Saginata, which is your Taenia. So in दोनों के eggs of these two organisms are acid fast. So CME, coccidian parasites, all three of them are acid fast. M is for Microsporidia, again acid fast, and E for eggs, two eggs of SS, Schistosoma mansoni and Saginata taenia, right? And then there are others. So what are the others? So there are spores of bacteria that are acid fast, and then there is the sperm head, which is acid fast, right? So here only I'm also going to talk about that how much percentage of the acid they are able to resist, right? So just remember it like this. The lepra bacilli requires 5% of sulfuric acid to resist, right? So lepra bacilli requires 5%. Legionella here requires 0.5 to 1%, right? Mycobacteria requires 20%, and nocardia requires 1%, right? So 5, 0.5 to 1, 20, and 1. That is how LMN, that is how you're going to remember it. All the parasites, this one is easy. all the parasites require just 1%. So 1%. So इसका क्या मतलब है कि जितना less acid it's able to resist, that means the level of mycolic acid is very, very less. So maximum mycolic acid is seen in mycobacteria because it's able to resist 20% of sulfuric acid, right? And then there are the spores. So can only spores can only resist 0.5 of sulfuric acid, right? And sperm head is also able to resist 0.5 to 1%. So just remember this table right here. So what are the structures? What are the organisms that are able to resist the acid fastness, and what percentage of sulfuric acid is required, right? So this is there. Next structure I've already talked about, and what is causing the acid fastness? This mycolic acid right here is causing the acid fastness. Okay? So this is all about your acid fast staining, and now I'm going to talk about the Zn stain. So what is a Zn stain? Zn stain is also known as a Ziehl-Neelsen stain, right? So it is the Ziehl-Neelsen stain, and it is known as a hot stain. Why is it known as a hot stain? Because we use heating here. So we have to use some form of heating here, right? So this is why it is known as a hot method of staining, which is Zn staining. You can get a question on this, that which method is the hot method of staining? It is the Zn staining, right? So what all are the stains that are required here? So the first stain, again, primary stain that we use in Zn staining, what is it? It is the carbonyl fusion, right? It is the carbonyl fusion that we use. So this one, carbonyl fusion, is a pink colored stain. This is the primary stain. Then we use heating. So after we have put the carbonyl fusion, we use heating. So what is this heating doing? This heating is actually making pores on the cell membrane. So this heating makes pores on membrane, and because of that, what is happening? This primary stain, so this stain is able to enter. Okay, so what is happening is we have put the primary stain and then we use heating. So this heating makes pores, and then those dye or this primary stain enters. And when we remove the heating, the stain is retained, the pores close, and the stain is retained. So that is why we require heating. Then we use a decolorizer. So what decolorizer are we using? As we spoke about, we use H2SO4, which is your sulfuric acid. Okay, so we use sulfuric acid as a decolorizer, and then finally we use a counter stain to stain the background. And what is the counter stain? We use methylene blue, right? So we use methylene blue. So let's see here. So what is happening here? So we add a primary. So these are going to be the steps. So add a primary stain of carbonyl fusion for 30 seconds. So we have kept it, and now what we are doing it? So this is actually kept for 5 minutes. It's not kept for 30 seconds. So for 5 minutes we have kept it, and now what is happening? We are applying heat. So when we are applying heat, what is happening? The cell membrane is opening, making pores, dye is going in. When we remove it from heating, the pores close and the stain is retained, right? Then we decolorize it by acid alcohol or sulfuric acid. So by that, what is going to happen? The ones that are acid fast, right? The organisms that are acid fast are going to resist it, right? So, and the other ones, other bacteria that are not acid fast are going to lose this stain. Okay, because they don't have mycolic acids in them to resist this acid fastness. And then we are going to add a counter stain, which is your methylene blue. So what is going to happen is the acid fast organism is going to appear as pink, and the other acid fast organisms are going to appear as blue. So your whole background and all the other organisms are going to look as blue, and only your acid fast structures are going to look as pink. So this is something that you see, right? Can you see this? So what is happening here? All these backgrounds are stained as blue because of the counter stain, which is your methylene blue, and all your acid fast structures, can you see these? These are stained pink. So these are pink bacilli against your blue background. So how do we report it? On whenever we want to report it in our exam, how do we want to report it? We report it as they are long, slender, beaded, right? They're long, slender, and beaded, straight to curved. ठीक है? They are straight to curved, long, slender, and beaded, straight to curve, pink to red bacilli against blue background. So that is what it is, right? That is how we are going to report it. And I've already spoken to you about the acid fastness. So जितना ज़्यादा mycolic acid होगा, right? जितना ज़्यादा mycolic acid होगा, उतना ज़्यादा it will be able to fastness उतनी ज़्यादा increase होगी और resist the fastness. ठीक है? So this is your Zn stain, which was your hot stain because we were applying some form of heat here. ठीक है? So this is your Zn staining. Then let's come to the Quignon's method. So what is happening in the Quignon stain? Here we are not using the heat. That is why it is known as a cold stain, right? That is why it is known as a cold stain. So what they we have made certain modifications here. So eventual outlook is the same. देखने में everything is the same, but here we are making some changes. So what are we doing here? It's a cold stain because no heat is required, right? So here what is the primary stain? Here the primary stain is carbon fission only. But what are we doing here? We are increasing the concentration. So we are increasing the concentration, right? Then the second one is the decolorizer because we are not heating. So now what we have to do something with the decolorizer. What are we doing? We are changing it from sulfuric acid to acid alcohol, right? So acid alcohol, what does this do? Even without the heating, this acid alcohol complex is going to form pores on the membrane, right? So because of this acid alcohol, they're going to form pores on the membrane. So primary stain में carbon fission डाला, concentration बढ़ा दी. Carbon, even Zn में भी primary stain is going to be carbon fission only, but here because we don't want to heat it, so we have increased the concentration, right? Second one is a decolorizer. There we were using sulfuric acid only. Here we are using an acid alcohol complex. So this acid alcohol complex is going to eliminate the heating and create pores on the membrane, right? And then we have our secondary stain or the counter stain. So here we can also use methylene blue or we can also use malachite green. So your end result is the same, but just this whole Quignon's method is a cold stain which is happening without heating. And this can also come as a question, ki what is the cold stain of checking the acid fastness of an organism? So that method is called as the cold Quignon's stain, right? The Quignon's method. So that is all about your acid fast organisms. Next stain that I'm going to talk about is going to be your Albert stain. So Albert staining किस organism के लिए करते हैं? Which organisms are mainly we use Albert stain for. So we use this for Corynebacterium, diphtheriae. And what is it differentiating from? So ये differential stain है, it's supposed to differentiate, right? So it differentiates between organisms that have metachromatic granules from those that don't. So mainly, which are the organisms that have metachromatic granules? So diphtheria has metachromatic granules, and which ones don't have metachromatic granules? The diphtheroids. Right, so that is how we are able to differentiate. So this differential stain actually differentiates between diphtheria and diphtheroids based upon the presence of the metachromatic granules, right? So what happens is, can you see here? So this is how your diphtheria is seen on a gram stain. It is a gram positive bacilli. Remember the McDonald's? right? Remember McDonald's, gram-positive bacilli, and this is what an Albert stain picture looks like. So Albert stain stains the bacteria green while staining the metachromatic granules as blue to black. So this particular picture will be seen in which? It will be seen in diphtheria, but it will not be seen in your diphtheroids. So this Albert stain is able to differentiate between diphtheria and diphtheroids based upon the presence of your metachromatic granules, right? So, so just remember these metachromatic granules. They are also known as, so these metachromatic granules are also known as Babes, Ernst granules or Volutin granules, right? So what are these metachromatic granules? They are actually the storage, like the food storage of the organism. So whenever the organism wants to store the food or the energy, it stores it in the form of these metachromatic granules. And this is very, very specific to diphtheria, right? So can you see all these ones at the end, poles? These one are the metachromatic granules. So this is going to be your Albert stain. So let's talk about the steps of the Albert stain. Albert stain is very, very simple. So firstly, before starting everything, we have fixed the slide. So the first step is going to be fixing the slide. So we have fixed the slide, and then what are we going to do? We are going to put Albert 1. So what does Albert 1 contain? So what are the two colors that are happening here? One is a blue color and one is a green color, right? So what is there? So Albert 1 we put for 5 minutes, and it contains what all? It contains toluidine blue, right? It contains your malachite green. It contains toluidine blue. It contains malachite green. Then what all does it contain? It contains glacial acetic acid, right? And then it contains your alcohol. So these are the components of Albert 1. So what do? We put Albert 1 for 1 minute, for 5 minutes, and then we wait. After 5 minutes, what is the next step? So we have to wash it. So we will just wash the slide, we will drain it, and then we will wash the slide. And then we are going to add Albert 2. So what does Albert 2 contain? Albert 2 contains iodine and potassium iodide. So again, what is this? Iodine is always working as what? It is working as a mordant. So whatever stain that the organism has retained, so they are going to fix that stain. So this Albert 2 is going to fix that, right? And it's not going to let that organism leave that stain. So what happens here? Because of the malachite green, the bacilli is looking as green, and because of the toluidine blue, the metachromatic granules are stained blue. But when it comes to your diphtheroids, what's going to happen? Diphtheroids doesn't have any metachromatic granules, right? So what it's going to be? It's going to just be green bacilli. So it's just going to be a green bacilli, right? So that is your Albert staining. That is your Albert staining. And all these stains, just remember, all of these staining methods are always seen. Just remember this, always seen under oil immersion. So what is oil immersion? It is under 100x microscope, right? 100x जो उसकी magnification होती है, उस microscope पे only we see all these stained specimens, right? Next, I'm going to talk about your bipolar staining. So what is actually bipolar staining? There are few organisms that stain just at the poles, right? So bipolar, bi means two and polar means at the poles. So the stainings are taking place at the poles. Can you see this photo right here? What's happening? Only at the poles it's staining, right? So this is also known as a safety pin appearance. So safety pin appearance because it's looking like a safety pin. Safety pin के ends पे ऐसे दो होते हैं ना, ऐसे होता है एक safety pin. So this is what is known as a bipolar appearance or safety pin appearance. And you just have to remember what all organisms give this kind of a safe tip in appearance. So then again, very, very famous mnemonic that we use for this bipolar staining is can produce very high yield, right? So can is for Campylobacter, P is for pseudo malai and malai. And these are species of what? These are species of Burkholderia. Right, these are species of Burkholderia. So Burkholderia pseudomallei and Burkholderia malai both give safe tip in appearance. V is for your Vibrio, but not the Vibrio cholerae that we're talking. Vibrio cholerae is not, safe, does not give the safe tip in appearance. But one species of Vibrio does. Which one is it? Vibrio parahemolyticus. Right, Vibrio parahemolyticus. Next we have H. H for Haemophilus ducreyi, right? H for Haemophilus ducreyi, and finally Y is for Yersinia pestis. So this particular mnemonic can produce very high yield. It's very, very important. You can get a photo of the safety pin appearance and they can say which organisms cause this kind of, show this kind of pattern. And you have to remember these 5 organisms. Which one are these? Campylobacter, Pseudomallei and Malai. They are two different, Pseudomallei and Malai. Then Vibrio parahemolyticus and Haemophilus ducreyi and Yersinia pestis. So these are the ones that show this kind of a safe tip in appearance, also known as bipolar staining, because they are staining at the poles, right? So this kind of completes my whole topic, and I'm going to now talk about certain previous year MCQs that have come from this particular topic, right? So especially related to nocardia, there are so many, this that has come. There are so many of the questions that have come. So identify the organism and staining method used in the provided image. So this particular question has come for almost 3 years, on 3 INICETs this has come. So which ones are these? They have given 4 of the options: nocardia acid fast staining, streptococcus gram staining, cryptococcus indiae, and coccidioidomycosis fast staining. So can you give me the answer? So this actually came in INICET in November 2023 and in 2017 and also in 2022. The same particular question has come. So what are we seeing here? So this is certain organism that is having some blue background, and then these are long, slender, pink colored organism. And which one is it going to be? Yes, the rain, very nice. It's going to be nocardia acid fast staining. It's definitely not streptococcus, because streptococcus is your cocci. These are not cocci. These are long, slender bacilli, right? Cryptococcus is a fungus. This is definitely not Indiae, because what did I tell you about Indiae? Indiae is black. We are not seeing a black background here. What background are we seeing? We are seeing a blue background here. So it's definitely not cryptococcus, definitely not streptococcus gram staining. Coccidioidomycosis past staining. Past staining can look something like this, but here the background is usually pink in color, right? And structures are stained pink, but coccidioidomycosis is again a structure like this. It's not a bacilli, it's not like long slender bacilli. So our answer here is going to be nocardia, right? So next, this is question number 2. Which of the followings are acid-fast staining organisms? And this question came in your INICET May 2022. And this was your question. They had given 5 organisms: nocardia, mycobacterium leprae, actinomycetes, cryptosporidium pavum, and isospora belli. And you had to guess what all organisms are acid-fast. So what are the organisms? What is the question? What is the answer going to be here? So the answer here is going to be B, right? The answer here is going to be B. Why? Because remember bacteria mein there was L, M and N. What was it? Leprae, nocardia and mycobacteria, right? Then in your parasites there were CME, coccidian, microsporidia and the eggs, right? And then you had spores. So what was missing? This actinomyces. This actinomyces is a gram-positive bacilli. It's not an acid-fast bacilli, right? So this one is an odd one here. Rest everything is an acid fast. So that is why 1, 2, 4, and 5. This option number B is the correct one, right? Then next, we are going to look at this question. So what is this question? A microbiologist stained a tissue sample and formed thin filamentous bacteria resembling Nocardia species. So now they've given the answer. It's the Nocardia species. So what staining technique should be used to confirm Nocardia? This is an INICET May 2022 question. So it is ZN stain, silver impregnation, Albert, or modified acid fast staining. So what is the answer going to be here? So the answer here is going to be D, which is your modified acid fast staining. Why? Because your Zn stain, the Zeal-Neelsen staining, is particularly said for your mycobacteria, which is your 20% H2SO4. Right? Anything that is modified beyond this, if we are making the H2SO4 1% or 2% or 0.5%, anything, so we call it a modified. So we have modified this sulfuric acid. It was supposed to be 20 because it was first invented for mycobacteria. But then we later realized that there are other organisms also that are acid fast, right? So what is happening here is because there are other organisms that are acid fast, so we have to modified this acid fast staining. So that is why anything beyond the 20% is called as a modified acid fast. So that is the answer that is going to be here, which is option number D, right? So yes, 1% is used. Very good. So that is going to be your answer here. So next, this is the question right here. So a 6-year-old boy complains of cough and throat pain. On examination of oral cavity, a white pseudomembrane was seen. Right, white pseudomembrane. So where are we going? In which direction are we going? He has throat pain. He has cough. He has, a pseudomembrane. Pseudomembrane is seen as which? See this. See this picture right here. What are we seeing? We are seeing gram-positive bacilli, right? So this is a gram staining. So what are the different stains that we use in a gram stain? Let's first see. Do we use an Albert stain? No. Do we use an acid alcohol? No. Do we use grams iodine? Yes. Do we use safranin? Yes. Do we use methylene blue? No, we don't. Do we use crystal violet? Yes, we do. Do we use carbon fission? No, we don't. So, do we use acetone? Yes, we do. So, we have marked 4 things. Now, let's see the sequence. What is the first thing that we use in a gram staining? We use the primary stain, which is the crystal violet. So, what is 6 is supposed to be the first one. So, everything else has been eliminated. So, this is going to be your answer. 6, 3, 8, and 4. Right? So, what is it going to be? It's going to be crystal violet, followed by iodine, followed by your acetone, followed by your safranin. Right? So that is how, that is what your answer is here. Next, we're going to talk about this question. A 32-year-old labor worker at a construction site presented with fever and hemoptysis. So he had fever and hemoptysis. Sputum sample was collected and smear shows the following. So this came in INICET May 2021 as well as 17. So this came twice. So what can you see here? So can you see there is some level of blue background seen here? So there is this blue background. And what else are you seeing? What structures are these? So these are definitely some acid fast structures, right? And what type of a method have they done? They have done, let's see the options. So gentian, violet, iodine, alcohol, and safranin. This is for gram staining, right? So this is definitely not the option. So this is for gram staining. Next, what? Methylene blue, malachite green, acetic acid, water. Can we see anything green here? Can we see anything green here? No, we can't. So this is definitely not going to be the answer. Next, we have methanol, methylene blue, acid, and water. Right? So methylene blue is there, methanol, acid, water. Let's keep this as an option. Next, what do we have? We have carbon fission, we have acid alcohol, and then we have methylene blue. So what is going to be? This is something which is your quinone stain. Right? So this is your quinone stain that we are using. We were not using methanol. We were using what? We were using the acid alcohol, right? That is the modification that we did. And we definitely had methylene blue and carbon fission. So our answer here is going to be D, right? So it's going to be D because we didn't mention anywhere methanol as being used as a decolorizer or as a, pore maker. So this is definitely not going to be there, right? So our answer here becomes D because this was Quignon stain that was done, right? Okay. So next we are going to talk about this question. So what is this question? A patient with fever and chills. So patient with fever and chills, throat, swollen glands, whitish membrane. Again, we are talking and going towards diphtheria, right? And Albert stain was performed. So we know Albert stain was performed for diphtheria, right? So now this one came in 2022, 20 and 2018, right? So, what are the constituents of Albert stain? So, remember Albert 1 and Albert 2. What was in Albert 1? Albert 1 had toluidine blue and malachite green. Did it have carbon fission? No, it didn't have carbon fission. Did it have methylene blue? No. No carbon fission, no malachite green. Toluidine and methylene blue, no. So what are the structures that are seen there? So we see a green bacilli, so malachite green, and we see blue metachromatic granules, so toluidine blue. So our answer here is going to be A. Yes, we use Quignon stain for isospora. It is just a method of using, another cold method for checking acid fastness. You can use Quignon method for any of these organisms, any of the acid fast organisms. Okay, so it's not just for the mycobacteria, it is for all the acid fast organisms, right? So yes, our answer here is going to be toluidine blue and malachite green because these are the ones that are there in the Albert one, right? Great. So now let's see about this. An HIV patient with fever. So we already know it's HIV, immunocompromised. It has fever, chronic diarrhea. So some diarrhea, some kind of a diarrhea. Coccidian, remember parasites, coccidian causing diarrhea, right? So stool showed oocysts. So just look at these oocysts. So these are the oocysts that are shown. They've also given you like a size, just to tell you, just to give you an idea of what it can be. Identify the components of the stain. So INICET, May 2019, this one came in. So what stain is it? It's definitely some form of an acid-fast staining because oocysts. Remember what parasites or what oocysts are acid fast? We talked about the three coccidian ones. First one being the Cryptosporidium, Cyclospora, and Cytoisospora, right? So this is definitely one of them, right? And this one here, it's going to be so what Cryptosporidium. Let me give you an answer. It's going to be Cryptosporidium, right? So identify the components of the stain. So let's see what all. So we have crystal violet. First question only crystal violet. Do we use crystal violet for acid fastness? No, we don't. So this one is definitely out. Again, like my last question, we are not using methanol in any of these stains. So this one is definitely out. Next, we have methylene blue, iodine, acid alcohol, and carbon fission. Are we using this? Were we using iodine here? Right? Are we using it? So this one is definitely not there. So what all are there? Sorry, just a second. Sorry, What are we using here? So again, methanol, quinyons, carbon fission, acid alcohol, methylene blue. Methanol are we using? We are not using methanol and acid alcohol both together. We are not doing that. So this is out. So our answer is actually going to be here. So we are using methylene blue. We did use iodine, acid alcohol, and carbon fission, right? So that is how what our answer is going to be. So that is it for the lecture. If you have any doubts, you can ask me anything about the staining techniques. Else, I'm done with the lecture and you can let me know. Any doubts? Yes, the order is wrong, that's it, but the components are the same, right? So, yeah, I think I'm done with the lecture. So, thank you everyone. And in case you have any doubts, you can message me and I'm happy to solve them. Okay, bye bye.